I'm Scott Miller. I've been involved in barcoding since the second Banbury conference. I should point out that Jonathan Coddington from the Smithsonian is also here. The barcode records in bold from the US is about almost 500,000. Although we know that there's a lot of barcode records that are not in bold, a lot of them sitting in people's hard drives and nowhere else. The United States, like some of the other countries we've talked about today, does not have an organized national barcode network. That in part is because of the diversity of the biodiversity research and management community in the United States, and it's really hard to sort of herd all of those cats, so to speak, because of the different governance and how many different kinds of organizations there are. A couple of trends that are interesting, though. We do see a lot more integration of DNA barcoding into ecological projects. Oftentimes they don't even call it DNA barcoding, but it's there. And we have put a lot of energy over the years into trying to get the United States government and some of the state natural resource regulatory agencies to use DNA barcoding as a standard. That's been with mixed success. But we do have some successes, like the Food and Drug Administration has something online called the Regulatory Fish Encyclopedia that includes voucher-based DNA barcodes. I just wanted to quickly talk about 3 projects, and the first 2 of these actually will be also mentioned in talks by Jeremy DeWard and Jonathan Coddington. Tomorrow, they're library construction projects based on broad collaboration. One of them is North American Lepidoptera. A few years ago, we noticed that there was like half of the North American species that were included in bold. This is butterflies and moths. So we set out on a path with Agriculture Canada and our U.S. Department of Agriculture colleagues and the Smithsonian and of course Guelph to actually fill those gaps, and we've been systematically collection mining at the Smithsonian collection. And as of today, we have actual sequences of 80% of the known Lepidoptera species for North America. And there's a lot more of them that are actually already a lot of the microleps are in the pipeline for NGS sequencing. So we will push it further than that. Then based on the success of that, and we figured out how to do this well with, at least with pinned dry insects. We decided to do sort of the same thing, but with arthropod, particularly insect genera, on a global basis to try to put more data points into bold to make it more useful for identifications. This particular project is funded by Jonathan Coddington's Global Genome Initiative project, and this is more of a startup enterprise, and a lot of these things have just been harvested but have not been actually sequenced yet. So we're making progress on that one. And then the last one that I will mention is basically an internal funding source for Smithsonian staff for DNA barcoding. Some of it's for library construction, some of it's for supporting research projects. We've been doing this for about 8 or so years. One of the things that we've noticed, well, a couple things. A lot of these are like marine invertebrates and various things that are actually pretty expensive to do because of the handling cost of getting good samples. But we've got about a little over 50,000 records that have been generated, but it's also in many cases it's been hard to get the researchers involved to liberate their sequences and make them public. And that leads me to the SWOT analysis. I think in the United States, both our strength and our weakness is that we have a diverse community. 3 threats that I will mention. One of them, like so many other people have mentioned, is just limited funding and lack of predictability of that funding. Then, as I mentioned, actually getting everybody involved in creating a sequence to agree to make the sequence public, even if they already agreed that they would do that. There's the issues of manuscript preparation, confirming identifications, permit issues, unclear data ownership, changing priorities, etc. A number of you are familiar with those issues, and it's something that will certainly be an issue for iBOL 2 and something that we might want to talk about tomorrow. And then the third one, which is not on that slide, but a number of other people have mentioned it, is Nagoya Protocol issues and those limitations. And I think one of the opportunities, which I will also talk about some tomorrow, is increasing use in ecological research and the intersection between library building and ecological analyses, and how we can sort of drive both of those forward in parallel. So that's it, dinner time.